fertilization and cleavage. Okay, what is fertilization? You know that very simple thing, fertilization, fusion of gametes, usually male and female gamete. Okay, now, so this will result in the formation of diploid zygote. Okay, that is known as fertilization, and this fertilization occur in ampulla, or may be written in examination or paper. You can see ampulla, isthmus, junction. Okay, but not both. They will not make you too much confused by giving these two options. Okay, there will be either ampulla or isthmus. Now, before fertilization, there should be some event known as pre-fertilization event. Okay, what will happen during these pre-fertilization events? There must be the arrival of sperm. During coitus, male discharge semen into female's vagina. That is the arrival of sperm. And the second is simultaneously the ovum is also released from ovary to ampulla. This is what? Fallopian tube. This is what? Ovary. And it is releasing the ova, and this ova will be transported through the help of membrane and ciliated epithelium to this junction. This junction will be known as ampulla isthmus junction, or that may be ampulla. Okay, second event is transport of ovum from ovary to ampulla. Now the third is very important. Okay, capacitation. This is what? Uterus. So whenever the sperm enters into the female body, it's need to be activated. What? Okay. Who will activate? Obviously, the sperm has almost activated by the prostaglandin and all things that is secreted in during the formation of sperm or semen. Okay. Now this sperm is helped by the female body. Okay, the female genital tract secrete some substance that help in the activation of sperm. Okay, the first thing is occur in what? Female body. Second thing is, what is the need of capacitation? Activation of sperm. How this activation will be done? By secretion of some substance. So what happens during capacitation? Okay, let me draw only the acrosomal or head portion of sperm. This is what condensed. This is acrosome. Okay, nucleus. Okay, this is the head. So there is some coating substance over this acrosome that can prevent fertilization. So the removal of this coating substance is necessary. Otherwise, how it will secrete this secretion? Hyaluronidase, neuraminidase, and zonalizin. So what is the another name of zonalizin? Acrosin. Okay, remember this. Clear? So what happens in the capacitation? That activation of sperm is done by secretion of substance that removes the coating of acrosome. Now this sperm is active to penetrate egg. Egg means secondary oocyte. Now we will discuss the last portion of pre-fertilization event. Now the sperm is ready to fertilize. Will secrete the acrosomal content. The acrosomal content consists of some hydrolytic enzyme like hyaluronidase, neuraminidase, and zonalizin. Okay. So this secretion is known as acrosomal reaction. Okay. So what happens in the acrosomal reaction? The sperm head fuses with outer membrane of ova. Okay, या फिर इसको भी रहने दो, है ना? This could be the last step. So we will start with the first step. Enzymes are released. First of all, enzymes are released. Okay, like hyaluronidase, zonalysin, and neuraminidase. And this will penetrate zona pellucida, है ना? So this is the acrosomal reaction. Now, the most important and the last topic for fertilization. The fertilization has following process. Number one, syngamy. ठीक है? So basically, this is the fusion of male and female gamete. Whenever the male and female gamete fuses, the process is known as syngamy. And there will be a mixing of cytoplasm. ठीक है? Plasmogamy. ठीक? Fusion of nucleus, progamy. ठीक है? The intermixing of chromosome, amphimix. That is very important points. Okay? So all these things occurs and this led to the formation of zygote. So as soon as the zygote is formed, what will happen? There will be a division, but this division will be different from the normal mitotic division. This division is known as cleavage. Okay, the fertilized egg or the activated egg goes for repeated cell division. Okay, and it will result in the formation of, if it is zygote, it will go like this: 2 cell, then 4 cell, okay, then 8 cell. Now this cell will try to arrange in a manner. Okay, so this repeated division of cell known as cleavage. And now, what is the difference between cleavage and mitosis? You can see here. First of all, there is increase in nucleus in case of karyoplasmic index. You can nucleus in case of cleavage, okay? But in case of mitosis, the karyoplasmic index remain constant, okay? But in case of cleavage, the karyoplasmic index increases. So very important thing, you must understand these cells which are being produced after division. This was the zygote, okay? The zygote gave rise to these cell. These cell will be known as blastomeres. Okay. Now, as these cells start dividing, okay, the mass of blastomere will remain equal, but the size will get decrease. The number will increase. Okay. The total size, I have drawn it differently, but total size of blastomere will be equal to the size of zygotes. And if they are, you can see here, they are multiplying or repeatedly producing. So there will be a very fast interface, because interface is a phase where the DNA synthesis occur. Okay, so every new cell requires DNA, so interface occur very fast and very short phase. Okay, you know that each cell is not growing in size but growing in number. Only nucleus is being increases. Okay, so what is interface consists of which phases? G1 as G2. G1 is for the cell mass increment, cell organelle duplication, and all these things. ATPs, RNAs, enzymes, etc. Okay. and G2 is for the remaining cell organelle duplicates to tubulin protein formation. Do they occur here? Size is constant, so the G1 and G2 will not occur. Only which phase occur? S phase. ठीक है?